But I'm going to be talking about the starch time bomb, breaking down starch breakdown. And I think today and even on Tuesday, starch has come up quite a few times. So it's quite a relevant topic. And I'm going to try to answer some questions. What is starch? Is starch an important factor in harvest maturity? Is this iodine test a reliable measure of starch? How quickly does starch break down in storage? Is it in days? Is it in weeks? Is it in months? What is the role of starch in fruit ripening and storage? And how does the rate of starch conversion to sugars affect the fruit quality? So these are all questions. But I think before we start, let's just ask ourselves, what is starch? Now, starch is a carbohydrate, we know that, and it consists of numerous glucose units joined by glycosidic bonds. So you get two types of molecules. You get the linear amylose, and you get the branched amylopectin. Okay, so you've got two main molecules of starch. Depending on the plant, starch generally contains about 25% amylose and 75% amylopectin by weight. Now, apples fall into this category. So how is starch produced in plants? I'm not going to go into the depth of the biochemistry of photosynthesis, but photosynthesis is the process of using energy from sunlight to make food, and that food is really the carbohydrates that we're referring to. It's a multi-step biochemical pathway, and it uses the energy of sunlight to fix carbon dioxide and to transfer that energy into carbohydrates, and in the process, it releases oxygen. So it's a very simple equation that you've all learnt at school. And here's two other things you would have learnt at school, the Calvin cycle and the Krebs cycle. But how are they relevant in the story? Well, the Calvin cycle takes place during photosynthesis, and it takes place in the chloroplasts. Energy is used along with carbon dioxide to produce these carbohydrates, as I mentioned earlier. And those carbohydrates will be in the form of glucose or sucrose or fructose and starch. The Krebs cycle, on the other hand, is a stage in respiration. And again, respiration is a topic that's come up several times during these talks. And this takes place in the mitochondria, where energy, or ATP, is now created. CO2 is produced, and it does this by breaking down carbohydrates. So it's the reverse of the Calvin cycle. So why is energy storage important in plants? Glucose is used to generate chemical energy, which is required for general metabolism. Okay, so it maintains the health of your cells. It's also the precursor to many organic building blocks, including nucleic acids, lipids, proteins, structural polysaccharides such as cellulose, and we know that's an important component of cell walls. So this carbohydrate can be used immediately for cellular respiration. So during photosynthesis, as it's created, the plant is growing. You need energy in order to maintain that tree. And so this is where this energy is used up immediately during respiration. However, any excess of that energy is stored now as starch. But the plant doesn't die at night. During night, photosynthesis stops. But it is that time that reserved energy in starch is still used to maintain cellular metabolism and maintain the integrity of those cells. Starch accumulates in developing fruit as sorbitol and converts mainly to fructose with glucose and sucrose. So just remember, and we've just heard about sorbitol now. So this is how it comes into the developing fruit. But now here we've got a ripening dilemma. We've got two different agendas. We've got the apple tree, and that's programmed to produce senescing fruit to provide viable seed. It's talking reproduction. It's talking survival. Whereas on the other hand, we've got the producer or the packhouse who wants to store fruit and delay the senescence as long as possible and yet still provide fruit of good eating quality at the end of that storage period. Once harvested, fruit then go into a state of decline. They leave the mothership. They lose the lifeblood of the tree, and they are what they are. They can't get any better than they are. The starch reserves that they've got are the starch reserves that they've got. So from that point on, they need to rely on that stored carbohydrate, which are the sugars and the starch, as an energy source to stay alive. So just remember that fruit are still living and respiring. Which then asks us the question, when is the best time to harvest fruit? Because to maximize storage life and fruit value, fruit must be harvested as close to ripe as possible, but with enough food reserves to survive the storage period until that marketing period is over and the fruit is consumed. Tree-ripe fruit, on the other hand, have the best flavor and eating quality, but they have low food reserves and have already started their decline. And obviously, these need to be harvested and marketed immediately. So in order to have a good fruit, you need to harvest earlier at an optimum maturity, but with sufficient food reserves to maintain fruit quality during storage. Now, I've mentioned respiration, and respiration is that process that keeps your fruit, your integrity of your cells through the release of energy, but it is also the process that results in the ripening of your fruit. So it's a bit of a balancing act. But in order to store your fruit, you need to slow down respiration, and that is what you're doing when you're storing it under CA, because under CA, you're dropping the oxygen. So on this side of the equation, you remove the oxygen, you're going to slow down respiration. On the other side of the equation, you can increase carbon dioxide, such as in modified atmosphere packaging or CA, and that is also going to have a negative effect on respiration. And likewise, if you remove heat energy by storing your fruit at low temperatures, that will also slow down respiration. So these are also very important points when it comes to storage quality. Now I'm going to ask the question, how important is starch as a maturity parameter? We know now that starch degrades during the fruit ripening to sugars. We also know that we use an iodine staining test or starch index, which is widely used as a maturity index. And we use this index to provide an estimation of starch content, provide a guidance to the level of fruit maturity, and also to give us an appropriate time to harvest for immediate consumption or for long-term storage. Hortec tracked this before you harvest your fruit. So if you're part of the maturity indexing program, you'll give fruits a month before, and Hortec will track your starch breakdown. Now, along with that data, they'll always give you a long-term average. And on either side of that long-term average, they'll give you the last 3 or 4 years. And you look at that data to try and work out how well you're doing. So if we look at Crips Pink in this season, and from the Algan area, we see up to about 196 days after full bloom, we're very close to the long-term average, and then we suddenly see a large rate of increase of starch conversion. So you can use this data by looking at previous years to similar profiles and saying, how did that fruit store? What can I learn from these ripening profiles? Jason showed this graph on Tuesday. He looked at Rosy Glow. We looked at 2 seasons. If you look at 23, there's quite from 5 weeks before harvest, there's quite a quick starch conversion compared to 2024, where it was different. Now, they both started at the same point. But the question then is, what effect does this difference in rate of change of starch breakdown have on your post-storage quality? Chris mentioned some pre-harvest ripening inhibitors, AVG and 1-MCP. We know that they also affect starch breakdown. I've just got an example here of Red Delicious and Granny Smith, where you get your typical starch breakdown of your untreated fruit, and that you see that the 1-MCP holds back that starch breakdown, as does the retain, although one gets differences depending on the different fruit types. Starch is measured with starch charts to give a starch index. Now, in South Africa, we've got two types of starch charts. We've got the circular type and the radial type. Chris, you'll be familiar with the canal starch chart, which has got a rating, index of 1 to 8, which is used mainly in the States. So, does the starch index relate well to starch content in starch tissues? Starch conversion, the test uses iodine and potassium iodide solution, and this reacts with long-chain amylose starch molecules to produce that dark blue color that you see. As that amylose is broken down into simple sugars, smaller carbohydrate units such as glucose, you don't get as much amylose, and therefore it doesn't stain as much. And that is the principle of your starch conversion. Now, we've also found, or we heard earlier, that amylose, however, only represents about 25% of your starch of your carbohydrate, your starch in your apple by weight. Which then asks the question, if 75% of your starch is amylopectin, and we know that this does not react as well as the amylose with the starch test, how accurate then is the starch index from the charts? We also see that you get different degradation patterns in the starch in the cortex, which is why we have the two different types of charts, the circular chart and the radial chart. And this may therefore be due to different ratios of amylose and amylopectin in the fruit. And we discussed on Tuesday about the gray color. You know, this could also relate to the gray color being part of that story. So as I mentioned, you get these two different starch charts where you've got either a radial type of pattern or a circular type, which is on the right-hand side. Now, while the starch index can give an indication of starch maturity, the interpretation has to be taken in context, because several biological and environmental factors may also influence starch levels, such as you get more starch in lighter crop trees. So your tree is still producing carbohydrates, but now there's fewer fruit, and so there's more starch going into that fruit, and so you're going to get a bigger starch or a darker color. Likewise, we know that there are cultivar differences. We know that Fuji has more amylopectin than maybe other cultivars, which gives you a lighter stain. Environmental stresses can also affect how much starch you get in your fruit, because during a warm summer night where the plants are respiring more, it will therefore use up more carbohydrates, which can also affect your staining. So one always needs to therefore look at other maturity indexes along with starch. In Experica, we did a trial looking at the rate of starch breakdown in apples, and we looked at Crips Pink and Crips Red. We harvested them, and we either treated them immediately with 1-MCP and then monitored the starch breakdown at ambient every 2 days, or we kept some fruit at -0.5 for 7 days and then looked at the rate of starch breakdown. And what did we find? Well, we found that if we look at Crips Pink, we harvest them quite late at about 45% starch breakdown, but within a couple of weeks, nearly all that starch was gone. We found that the 1-MCP did hold this back slightly, as we'd expect, and after 7 days, we saw a very similar pattern. But what was interesting, within those 7 days, we got a 20% starch breakdown. And if you take these 2 points and you extrapolate it into a linear line, because it's a linear breakdown, you can get an equation from that line, and from that equation we can calculate that there'd probably be very little starch left under -0.5 within 6 weeks of storage. Likewise, with the Crips Red, we saw a very similar effect, except now we harvested at 25% starch breakdown. So the fruit were a little bit less mature. We saw a better response with the 1-MCP, so the divergence of the two lines was more pronounced. And after 7 days, we found that there was only a 10% starch difference. So the earlier you harvest your fruit, the slower the rate of change of starch is. In other words, the slower it breaks down. So that is why starch breakdown is so important, and it's why it's important to get the correct harvest maturity. And in this case, if we do the same exercises with the Cribs Pink, we see that with the graph, it'll take now 10 weeks, almost twice as long, and you get a full starch breakdown probably between 10 and 12 weeks. And we asked ourselves, have other people also found this information? And we looked at a paper by Niewald, Streif, and Kitterman, and at ambient, they found within just over 2 weeks that nearly all the starch was gone, and at a lower temperature, it held back that starch breakdown. So the question arises, if carbohydrate or the starch is used up within 2 or so months of storage, how does that fruit maintain its quality? So we asked ourselves, well, there must be other starch reserves that are not stained by iodine, and we know that that's sort of the amylopectin. So we said, well, then let's look at starch content, because starch content will include both of those molecules. We're still waiting for our results, so then I went into the literature, and I found a paper by Tamawong and Arakawa, where they looked at the starch degradation on detached apples. They looked at 2 cultivars, Sagura and Fuji. The Sagura is an early maturing, faster respiring apple compared to Fuji. They're harvesting them either immature or mature, and then they treated them either with ethylene or 1-MCP. Ripened at 25 degrees, and then they measured the starch content. So what did they find? Well, they found that after the fruit were harvested, within 8 days, the immature fruit practically had no starch left. And the mature fruit had a slower rate of change of breakdown, but within about 12 days, and maybe a little bit later, there was still no starch in the fruit. They also found that 1-MCP did slow down this breakdown, as we found in our trial. So if you look at the Fuji, and this is a slightly slower respiring apple, maturing apple, again, they found practically no starch breakdown within about 2 weeks of storage. So this is makes one think. And again, the 1-MCP did have an effect, but in the end, it didn't have a major effect that it would prevent the starch breakdown. They also had a paper by the same authors looking at starch degradation where they looked at fruit harvested from a tree over several different harvests, the same 2 apples, but now they harvested at 5 or 6 stages, different days after full bloom, and they looked at the maturity, the starch content, and the sugar content. So if we look at this graph, you can see then that the first graph represents the Sagaru, which is the faster respiring fruit. But what was quite interesting is there was very little starch breakdown until about 110 days after full bloom, and then suddenly the next time they measured the fruit, there was practically no starch. And just remember that these fruit were harvested 1 week before commercial harvest. Now, the Asian prefer their fruit very sweet, so they're not going to harvest fruit for long-term storage. So just bear that in mind. So this isn't really a South African scenario, but the principles remain the same. Likewise with the Fuji, which are also harvested a week before commercial harvest, you got a slightly different starch breakdown pattern. You got a slower, more regular starch breakdown, and whereas the Tsuguru took 55 days, the Fuji took 75 days to reach the same point. If we then look at these two, the 2 top graphs is, the first one is amylose and then amylopectin for the Tsugaru, and likewise for the Fuji on the bottom, you find that point where they were not getting any starch, if they looked at starch content in terms of amylose and amylopectin, again, there was nothing left in the fruit. It was basically all gone. So what happens to this starch? Well, we know that it goes into sugars, and that's exactly what they found, is that that there was, as the starch broke down, you got more sugars in your fruit. So now, if we look at this and say, okay, what is this information telling us? Okay, from a post-harvest perspective, it tells us that starch is a vital energy source, okay, which is needed to maintain fruit quality. And research shows that that the starch is rapidly converted to sugars once harvested. Now, I don't know about you, but I was under the impression that your starch stayed around in your fruit for months. It doesn't seem to be the case. And we found it in our labs. We don't even bother to measure starch in a post-storage trial because we know there's nothing there. But I've never really thought about it. So those sugars are what's left then to produce the energy to maintain the integrity of those cells. We see that cold storage and 1MC can reduce that rate of starch breakdown, so they're both important tools. We know that the sugar increases during storage and through the respiration is used to provide energy for cellular maintenance. So as long as you've got the sugars in a form that can be used to maintain the integrity of your fruit and the metabolic processes continue, you will still have an edible fruit. But when it reaches that last stage of fruit development, which is now senescence, and this now involves those deuterative processes, and that's when you start getting the disorders. So sometimes we are actually taking our fruit out of cold storage at stage 4, at that last stage where you now are starting to pick up these problems. So what's important is to harvest fruit at the correct maturity. We see that the earlier you harvest it, the better chance you've got of holding back that starch breakdown. And using those post-harvest tools such as CA and 1-MCP and cooling to slow down respiration, which will then delay senescence and prolong the storage quality. For further research, when I started looking at this talk, I really went into the literature thinking there would be so much information on starch breakdown during cold storage, and I could not find much at all. And especially from a South African perspective, I'd love to assess the rate of starch conversion of cold store fruit over long-term cold storage, looking at different cultivars under different seasons, but also looking at the starch index and starch content and comparing them to sugars under different storage conditions, and then correlating this all to fruit quality. Because if you remember those early graphs where you can start predicting when you're going to have no starch breakdown, maybe one can develop a tool that can predict your quality from that. And that's my story. Thank you.